Hello everyone, this is Dr. Kalpana and you're watching my YouTube channel Pharmacy Lessons Made Easy. See, for the preparation of GPAT, I'm putting some important topics here so that you can emphasizely study these topics which are very much important from GPAT point of view. Although you have to study all the topics, but these are quite important from exam point of view. So let's begin. In this video, we shall be covering the important topics which are or which may be asked from the tablets and capsules dosage form, that is the solid dosage forms. First thing is the types of tablets. Like, you must be aware of what are compressed tablets, molded tablets or triturates. Okay, compressed like coated tablets, uncoated tablets, multiple compressed layer tablets or solution tablet, dispersible tablets, effervescent tablets, lozenges, chewable tablets, buccal and sublingual tablets, and the difference among them. Okay, so that is very much important, how a buccal tablet is different from lozenges. So buccal tablet basically is which is kept at the side of cheek or the buccal cavity for the slow release of medicament, except in the case of nitroglycerine and mannitol. Hexanitrate tablets we should dissolve in 1 to 2 minutes to produce the immediate effect. Okay, the chewable tablets in which mannitol is generally used as a base, which are required to be broken and chewed in between the teeth before ingestion. Okay, so it is for those who have difficulty in swallowing. Okay, and for those adults who dislike swallowing. Lozenges are the tablets that should not disintegrate in the oral cavity but should dissolve slowly in the mouth. Like, you keep Hanitus or Vicks tablet inside your mouth, that is called lozenges. Basically, there is no disintegrating agent that is included in the lozenges. So you must be aware about the formulation aspect, how that particular tablet is different in composition or preparation point of view. The tablets like soluble tablets which are required to dissolve completely in the liquid so as to produce the solution of a certain concentration, like mouthwash, gargles tablet, that way. Okay, all the ingredients used in the formulation of soluble tablets must be completely soluble in the solvent, otherwise that will not be considered as soluble tablet. If we talk about effervescent tablets, it has the major ingredients like sodium bicarbonate, citric acid and tartaric acid. In the presence of water, they will liberate carbon dioxide and produce effervescence. That is why they are called effervescent. Why we are making it? Because that will lead to disintegration of the tablet. Effervescence that is being produced due to the reaction of sodium bicarbonate and these acids by the action of water. And this will increase the palatability of the tablet. Okay, next thing is inserts or vaginal tablets. Some vaginal suppositories or pessaries are prepared by compression. Okay, they are also called they contain basically antibiotic or steroid. These are called inserts or vaginal tablets. Some laxative suppositories are also prepared using it. Okay, then implants, if we talk about, they have to be implanted intramuscularly or subcutaneously with a surgical cut into the skin. Okay, because we are dealing with surgery, so whatever you are doing as an operation or something like that, everything should be done aseptically, otherwise there would be chances of infection. And it should be packed in a unit dose and sterile container, right? If we talk about enteric coated tablets, they are different because they have to be disintegrated in the intestine and not in the stomach. That is why it is called enteric coated. Why we use it? Because such drugs are supposed to dissolve and disintegrate in the intestine only because of the pH differences. And this was a major categories. Like, if we talk about coated tablets, it may be either film coated by coating or with a water soluble material, like which will protect it from the atmospheric conditions. If we talk about sugar coated tablets, the main aim is to mask the objectionable taste or odor of that particular compound or the API. And if we talk about layered tablet, the granules of incompatible substances which are not compatible with each other, they are being compressed in the form of many layers, two or more, right? So obviously for preparing layer tablets, you should need a different type of machinery. Okay, this was about the types of tablets. Next thing you have to look upon is the tablet components, not only the API but which diluents are used, which type of diluents are directly compressible, okay, and what binders, at what concentration it should be utilized, different disintegrants, in which tablet disintegrant is added, in which tablet it is not, and if added at what concentration, what are the different compounds that are added, what are their trade names, like Avicel, that is microcrystalline cellulose, if it is added up to 10%, then it acts as a tablet disintegrant. The same way S-dicyl at 2% is used as tablet disintegrant, it is actually cross caramelose sodium, right? Then the glidants, like talc or colloidal silica. Talc is used generally at the concentration of 1 to 2%, and it is insoluble but not hydrophobic in nature. The colloidal silica is available in various grades like aerosil, cabosil. Okay, it's an excellent glidant. It is used in the concentration of 0.1 to 0.5%. Not only glidant but anti-adherents and lubricants are also added to the tablet formation as an ingredient in a very minute quantity, like lubricants like sodium stearyl fumarate or macrogols or magnesium lauryl sulfate, liquid paraffin. Okay, they can be added at different concentrations. The main aim would be making something water soluble or as a as wetting agent. Okay, and anti-adherents are basically required to reduce the sticking or adhesion, like talc or magnesium stearate or starch derivatives. They are generally used. FD and Sifu and D and C dyes are basically used as colors. Flavors, the maximum limit up to 0.5 to 0.75% concentration, they are also added. And if we talk about sweeteners to make it chewable, in the case of chewable tablets, you can use saccharin, mannitol, aspartame. Okay, and then you have to look upon the defects. They are the most important ones. Okay, from GPAT point of view, you must be aware of the different defects during tablet formation. Like most important to name a few are capping and lamination. It is generally partial or complete separation of top or bottom crown of the tablet from the main body of tablet. Okay, it may take place due to air entrapment or deep concave punches, and the percentage of moisture also plays an important role in it. Another way may be incorrect setup of the machine, the rotary tablet machine, or the way where actually the compression cycle is to be carried out. Okay, so this was about like capping and lamination. Next one, like another important one, picking and sticking. That means surface material from a tablet which is sticking to and being removed from the tablet surface by a punch is called picking. It can be taking place when the tips of the punch are either engraved or embossed. Okay, especially for the letters like O, A, and B, enclosed area which is found, it is very difficult to get manufactured very clearly. Okay, so this was about picking. And sticking is the tablet material which will stick or adhere to the die wall. Okay, so to prevent such damages or defects like picking and sticking, as large as possible letters should be made for in font size, okay, particularly the punch with small diameters, right? Moreover, if you plate the faces of the punch with chromium, that will also produce a smooth and an adherent face, so that will also prevent picking and sticking. Another important defect in the tablet is molting. Molting is an unequal distribution of color on the tablet. This is very frequently asked question for GPAT. What is mottling? Okay, so that will, because of the unequal distribution, the light and dark areas would be graded. Otherwise, the surface would be uniform in color. So, to make a dye uniformly dispersed, you can change the solvent system or the binder system, or change the temperature at which the tablet is getting dried, or you can even choose the smaller particle size that is being compressed for tablet formation. Another important defect is double impression. That means the in the condition like lower punch is somehow in the free condition and is uncontrolled for a small distance and drop down before it rides up for the ejection, then the ejection arm will push the tablet out of the die. During this so-called short distance where it is free to drop or free to travel, it rotates. So in spite of single impression, it will show the double impression onto the tablet surface. So such problems can be encountered if there are upper punch engraved form are in the engraved form, or the tablet machine utilize two compression stages for compressing the tablet. Okay, so these are the important topic of defects of tablet. Another very important aspect is the coating of tablet coating. Okay, what are the different compositions of the tablet? Why do we do it? Basically, for the sugar coating, for masking the odor and the taste of the particular API, and in the case of film coating, to protect that particular tablet from damage from the environmental conditions. Okay, and enteric coated so as to get disintegration or the effect at a particular site, especially in the intestine, not in the stomach. So that is a basic purpose, and the composition is actually the main component, like film former. Otherwise the film will not form in the case of film coating. Film formers can be like HPMC, hydroxypropyl methyl cellulose, acrylate polymers, polyethylene glycols, povidone, hydroxypropyl cellulose, ethyl cellulose, methyl hydroxycellulose, such kind of materials. In the case of enteric coating, the materials like PVAP, okay, polyvinyl acetate phthalate, this is the most commonly used polymer film forming material. Hydroxypropyl methyl cellulose phthalate, HPMCP phthalate is a group which is added. Okay, similar way cellulose acetate phthalate, okay, its short form is cap. Okay, that is also an example of enteric coated material, along with the acrylate polymers like Udragit L and S. Okay, so these are the enteric materials. If we talk about the basic components, then along with the film forming agents, there would be plasticizers like propylene glycol, glycerine, castor oil, organic esters, spans, tweens. Okay, there would be colorants, there would be matching of color and the flavor, also like caramel, carotenoids, flavones, chlorophyll, indigo, turmeric. These all are the colorants that are added actually. And in case of your sugar coating, the different steps, they are also very much important from exam point of view, GPAT point of view, because the different steps have to be followed for sugar coating. The basic steps are sealing, subcoating, syrping or smoothing, that is also called smoothing, finishing and polishing. In the seal coating, basically it is done to avoid the moisture penetration into the core of the tablet. Okay, and without this step the overwetted tablet would absorb excess quantity of water vapor or the moisture. This will lead to the disintegration and softening of the tablet, and obviously its physical and chemical stability would be affected. It can be possible to adjust the subcoats and further coats so that localized overwetting should not take place. Okay, the effective sealant that is used for this step, that is seal coating step, is shellac, zine. Okay, shellac although is very effective, but tablet disintegration and dissolution time becomes more on aging because of its polymerization. And if we talk about zine, it is an alcohol soluble protein which is derived from corn. Okay, and has also been used as a very effective sealant for this very first step, that is seal coating, done to prevent the moisture content. Okay, the next step in the sugar coating is subcoating. It is basically applied to round the edges, and which this is the most important question which is asked for the subcoating part is कि which step is used in the sugar coating which increases the tablet weight up to 100%. Okay, the answer is subcoating step, second step after the sealing step. The subcoating step is the step which will increase the weight of the tablet by 100%, 50 to 100%. Okay, the what is actually done in this step is alternatively you apply a sticky binder solution and then apply then do dusting of subcoating powder, then you have to subject it to drying. Okay, similarly subsequent subcoats are also applied in the same manner until the tablet edges have been covered and you achieve the desired thickness. Okay, so this was about the second step. First was sealing step, second is subcoating step. The third step is the syrup coating. The purpose of this step is to fill the imperfections, to cover those imperfections which might have been caused by subcoating, because you are repeatedly applying the solution, binder solution, then dusting, then drying. So this may make the surface of the tablet very rough. So to avoid that, syrup coating is done, and it will require the skills of a formulator. So first syrup coating is done containing some suspended powder and are called grossing syrups. Okay, the dilute coloring agent can also be added in this phase. So the question that might be asked in this for this step is, in which step you can add the coloring agent in sugar coating? So the correct answer is syrup coating. Okay, so this will facilitate the uniform coloring, right? The fourth step is the polishing step. This is done to achieve the required luster. Okay, after the sugar coating process. How it is done? Actually, the tablets that are to be polished in clean coating pans or canvas lined polishing pans are carefully applied with beeswax or carnauba wax. Okay, and then warm solution of these waxes in naphtha or suitable volatile solvents. Right, so the treatment of naphtha or these waxes is usually done in the polishing step. Right, so this was about the different types of coatings and the materials which are used and the steps to be followed for enteric coating or the film coating or sugar coating. Right, the another important thing which is asked is like the coating defect, like the tablet defects, the coating steps are also very much important. Okay, and in this also the sticking and picking is the defect. Second defect is the roughness. That means once the coating is applied by a spray, some droplets will dry very rapidly before reaching to the tablet bed, which will result in the formation of deposits on the surface dried particles, whereas the finely divided droplets will not be formed. Okay, another thing, the very important question that is asked is orange peel effect. Actually, once you are applying the coating solution, if it is not adequately spread before the drying, this may lead to orange peel or bumpy orange effect. Okay, that means uh, the spreading is either done by too rapid drying, or the solution was having very high viscosity. So if you dilute the solution with additional solvent, that may be the solution of this problem. So this is the orange peel effect, which is the most important one and asked in the GPAT exam. Another important one is bridging and filling. This is also tablet coating effect. Okay, what happens is during drying, the film which is being coated gets shrunken and pulled away from the very sharp corners of or the bisects, which results in the bridging of the surface impression. Okay, so it represent in it represent the bridge. Okay, or along the bisect, that line which converted into two different tablets. Okay, or tablet pieces, so that is called bridge formation. So if you increase the plasticizer content or you change the nature of the plasticizer, this may help out solving the problem of bridging. Filling is actually caused by applying so much narrow the monogram or bisect. In addition, if solution is applied very fastly, overwetting may lead to quick fill liquid, and that will be retained in the monogram. So if you cautiously monitor the fluid application rate and thorough mixing of the tablets in the pan, that will prevent the filling. Okay, another very important one is blistering. So this is also, blistering is also a tablet coating defect. Orange peel is coating defect. Bridging and filling is coating defect. Sticking and picking are both tablet defect as well as tablet coating defect, right? Blistering is when coated tablet need more drying in the oven, then too rapid evaporation from the surface of the solvent lead to affect the high temperature on strength and elasticity and adhesion of the film. It may result in the blistering. Okay, if you keep the drying conditions at moderate rate, then this problem can be sorted out. Another very important one is cracking. Cracking is also a tablet coating defect where internal stress in the film overpower the tensile strength of the film. So the tensile strength, if we increase it by using high molecular weight polymer or taking mixture of polymers, then this cracking defect can be overcome. Moreover, the color variation or hazing or dull film are also some important defects of tablet coating, right? Another most important topic is evaluation of tablets. So for this, you have to learn the limits. That is the most important thing. Like, what is the limit for the content of active ingredient in the tablet, in which condition it would pass the test and in which condition it would fail the test. Like in the case of 0.12 gram or less weight of each medicament, then you have to take the average value, limit less than 90 or greater than 110%. That is the basic limit, but you can subtract the lower limit of samples of 15, 10 and 5 samples by an amount. Like if it the tablet is having 0.12 gram or less quantity, then you can subtract the lower limit of 15 samples by 0.2, 10 samples by 0.7, and 5 sample by 1.6, and you can add the upper limit of the samples of 15 sample size by 0.3, 0.8, and 1.8 in respectively in case of 15, 10, and 5 samples. So that way you have to learn the limits in which condition how many tablets are required, like 20 tablets are required for this weight variation test. Next most important test is the uniformity of weight test. That means the uncoated tablet should comply with 20 tablets weight. This is that 20 tablets should be selected at random, and then you calculate the average weight. In the case not more than 2 of the individual weight should deviate from the average weight by more than the percent deviation as described by the pharmacopoeia, right? So for average weight of tablet 80 mg or less than 80 mg, the percent deviation that is allowed is 10%. If the average weight of tablet is more than 80 mg and less than 250 mg, then the percent deviation that is allowed is 7.5%. If 250 mg or more than that, then 5% deviation is allowed, right? The same way size, shape, organoleptic property or unique identification mark, uniformity of content, they are not that much important. The hardness by what instrument it is test, like Monsanto tablet hardness, strong cope tester, or Pfizer tester, Eureka tester, by which the hardness test is determined, Schullinger tester, that are the fibrilators basically that operate in the horizontal position and is used to determine the acceptable or unacceptable limit of the friability, which is an index of tablet strength. The laboratory friability test is known as Roche fibrillator, is a plastic chamber which actually rotate for 100 rotations for like for at if it is revolving at 25 rpm and dropping the tablet at a distance of 6 inches with each revolution, then pre-weighed tablet samples are placed in the fibrillator, which is then operated for 100 revolution, that means for 4 minutes. The tablet are then dusted and reweighed. So the tablet should not show a variation in the weight by more than 0.5 to 1%, okay, then it would be acceptable, otherwise will not accept it. Okay, and in the case of disintegration test and the dissolution test, for different types of different types of tablets, the disintegration and dissolution limits are different, so you must be aware of for the different types of tablets. Okay, so if it is a chewable tablet, then no disintegration is required. Coated tablet except the chewable tablet should comply with disintegration test. Okay, and IP apparatus, you must be aware of the limits. Enteric coated tablets should comply with DT test of enteric coated tablets. Okay, soluble tablet, dispersible tablet disintegrate within 3 minutes when examined by the disintegration test for tablet using water at 19 to 21 degrees Celsius unless otherwise stated in the individual monograph of that particular tablet. Okay, so the limits that are being described in pharmacopoeia for different types of tablets, like for uncoated tablet, unless otherwise indicated in the individual monograph, one tablet each should be placed in the 6 tubes of the basket for disintegration test, and the disc is to be added in each tube after operating the apparatus at the described water limit, that the temperature limit, that is 37 plus minus 2 degrees Celsius. At the end of 15 minutes you have to observe the tablets. If all the 6 tablets would be disintegrated, then the tablet would pass the test. If one or two tablet fail to do so, then the test has to be repeated on 12 additional tablets. That means total 18 tablets are there. First you have done the test for 6, then if one or two tablets fail to show the disintegration, then you have to do it on 12 additional, so total would be 18. So after repeating the test, if not less than 16 of the total 18 tablets have disintegrated, then it will be, the test will be passed. Okay, in the case of coated tablets, all the other conditions would remain the same. If any tablet have not disintegrated, you have to repeat the test on further 6 tablets using water with 0.1 normal hydrochloric acid. The tablet then pass the test if all the 6 tablets have disintegrated in the acid medium. If one or two tablets still fail to disintegrate, then you have to repeat the test on 12 additional tablets. Not less than 16 out of the total 18 should fully disintegrate in that condition. In case of enteric coated, the same apparatus has to be operated first with 0.1 normal HCl maintained at 37 plus minus 2, and no tablet should show the sign of disintegration under that condition. Then you have to operate the same apparatus using mixed phosphate buffer pH 6.8 by maintaining the temperature at 37 plus minus 2 degree. At the end of 60 minutes, you have to remove the assembly from the liquid and observe the tablets. Right? If you have to do the same test for capsules, then the capsule should be placed and maintained at 37 plus minus 2 degree Celsius. If the hard capsule float on the top of water, then you have to omit the disc. Okay, while testing soft capsule, you can use the disc that is utilized for DT purpose. Okay, if two or not more than two, if one or two capsule fail, then you have to repeat on the 12 additional, and out of 18 total 16 should pass the test. So this was about the DT. Similar way, disintegration test is carried out for tablets and capsules, and the interpretation would be for each 5 tablets or capsules you have tested. In those cases, when one tablet or one capsule is directly to be placed in the basket for each test, the amount of API is not less than 70% of the stated amount. If two or more tablets or capsules are directly to be placed together in the basket for each test, in 5 replicate tests, the amount of API per tablet or capsule in each test should not be less than 70% of the stated amount unless otherwise specified. Right? The another one you have to look upon is the powders and its classification. The coarse powder, moderately coarse powder, moderately fine powder, very fine powder, microfine powder, super fine powder. That is very much well described in one of my video, which is based on size separation methods and the different categories or specifications of powders and sieves. Okay, that is also an important topic from GPAD point of view, right? So I think this would be enough for tablets. If we talk about capsules, then the important topic would be different types of gelatin. Okay, gelatin is the substance which is of natural origin but does not occur much in nature. It is actually prepared by hydrolysis of collagen, the main protein of connective tissue. Okay, the animal bones and skins are raw material for its manufacturing. There are mainly two types, type A gelatin and type B gelatin. Type A is produced by acid hydrolysis. Okay, the choice of manufacturing method depend on the nature of raw material. The skin are mainly acid processed, whereas bones are base processed and are called type B. Okay, the properties of gelatin are most important for capsule manufacturing. The most important properties are number one, its gel strength, that is called gel rigidity or bloom strength. Okay, and it is expressed as the loads in grams which are required to push a standard plunger a set distance into a prepared gelatin gel using 6.6% solution at 10 degrees Celsius. The gelatin used in hard capsule manufacture is termed high bloom gelatin, whereas soft capsule lower bloom material 150 to 200 bloom should be used. The viscosities of the gelatin solution is used to control the thickness of film or sheet that is being produced by the capsules. Okay, so the type of gelatin, then the composition of capsule, like soft or hard, both. uh, the composition, how they are different. The basic component which is added along with the gelatin is the plasticizer, which will make the film, gelatin film, more soft or flexible. So the quantity will decide the softness or hardness of the gelatin, the quantity of plasticizer. Okay, uh, if plasticizer is used in high proportion, that it will make it more softer. The most frequently used plasticizers are glycerol, sucrose, acacia, propylene glycol, sorbitol, glycerol. Then the third component along with gelatin plasticizer is colorant and preservative. The most commonly used colorants are either soluble dyes or insoluble pigments. Pigments may be used in the large quantity, like titanium dioxide, which is white in color and used as an opacifying agent. Other one are the pigments are iron oxides, like black, red, and yellow color. Okay, the colorants can be used, and it depends from country to country. Also, bicolor soft gelatin capsule aluminum flakes are used to prevent the color transfer among the two layers of the capsules. Moreover, these preservatives are used, right? The molecular weight of gelatin varies from 20,000 to 2 lakh, and the average molecular weight will depend the different grades of gelatin, like Pharmagel A and B. Pharmagel A has pH from 4 to 4.8 to 5.2, and B have pH from 6.5 to 9.5. Actually, it is a mixture of pork skin and bone that will give the shape to capsules. Pork skin gelatin contributes to plasticity, while bone gelatin gives firmness to the capsule. The preservative, if included, are generally the mixture of propyl paraben and methyl paraben. Propyl paraben is taken 1 part, and 4 part of methyl paraben are taken, and the total extent to which preservative is added is not more than 0.2%. Flavors should not exceed 2%, and sugars generally is used up to 5%. If we talk about the machinery for this, dipping followed by cold air circulation is the main mechanism by which gelatin capsules are shells are produced, and they should be stored under controlled condition. The normal moisture content of the shell is 10 to 15%. Okay, in the shells for human use are marketed at 8 size. The largest size of capsule is 000, and the smallest capsule size is number 5. For veterinary use, the large capsule numbers like 10, 11, 12 with approximate capacity of 30, 15, and 7.5 grams are also marketed. The material which is filled inside the hard capsules include the diluent, the protective sorbents, glidant, and anti-dusting compounds. Okay, the substances which are highly soluble in water, like citric acid, sodium chloride, ammonium compounds, they should not be filled in the gelatin capsules, because that may affect the gelatin shell, leading to abstraction of water from it. Moreover, some water soluble substance may also migrate into the gelatin shell that reduce the potency of the encapsulated material, like thiamine hydrochloride and ascorbic acid. The main steps are the filling and then finishing, followed by the dusting or polishing step. Can be like salt polishing, cloth polishing, brushing, dusting, and inspection is final step, which is done to pick up the imperfect or damaged capsules. For that purpose, rota sort is a sorting machine which is sold by Eli Lilly company. Okay. And the hand operated as well as the material filling equipment like rotofill is used as a filling equipment for capsules. There are some difficulties which are there in filling of capsule, like especially for deliquescent or hygroscopic powders, eutectic mixtures. So to prevent that, inert powders can be added while filling the granular powder. Also, the soft gelatin capsules are slightly different from the hard gelatin capsules, especially in the content of the plasticizer. If we talk about concentration, it can be expressed as a part of dry plasticizer to one part of dry gelatin. In practice, the ratio varies widely between zero point 0.3 to 1.0. This will impact the diversity of the plasticizer and film material. The low ratio is like zero point 0.3 and zero point 0.5 is used for oily liquid fills, between zero point 0.4 and zero point 0.6 for oily fills with added surfactant, and zero point 0.6 to 1.0 for water miscible fills and chewable capsules. Water generally range from zero point 0.7 to 1.3 part to each part of dry gelatin. Preservative that is used traditionally to prevent mold growth in the gelatin shell, mainly the common additives like ethyl and propyl hydroxybenzoate, methyl ethyl propyl paraben, these are used. Water soluble dyes are added as color, and titanium dioxide is a main opacifier which is used in soft gelatin capsule at the concentration from 0 to 0.5%. Then if you want to make it enteric coated, then you have to coat it with 4% cap, that is cellulose acetate phthalate, which we discussed. The soft gelatin capsules can be prepared by either plate process or rotary die process, which was developed by Robert P. Scherer. Norton capsule machine is actually fully automatic. Ecogel and Stern machine can fill the powders as well as the liquids in the soft gelatin capsules. Another important thing is base absorption value and the bloom strength, as we discussed earlier. Base absorption is basically defined as a number of gram of liquid base which is required so as to produce a mixture which will be encapsulated with 1 gram of solid. The value of base absorption can be obtained if we divide the weight of base by weight of solid. The formula that is used is Ba plus S into V upon D is equal to minimum per gram factor. That is the weight of liquid base required to make a capsulable base with 1 gram of solid. V stands for the volume and D for the density of the mixture. Minimum per gram factor will help the rapid calculation of the size of the capsule, and it will avoid the preparation of many possible mixtures in the laboratory. Bloom strength or gel strength is a measure of cohesive strength of the cross linking among the molecules of gelatin, and the molecular weight is directly proportional to this bloom strength. The method that is utilized to determine bloom strength is you have to take a plastic plunger and you have to make it fall down in a gelatin gel whose strength is to be determined. Generally, 6.67% of gelatin gel is held at 10 degree for 17 hours, and the plunger which is to be plunged inside this gel should be 0.5 inch in diameter and 4 in the thickness. So by this means the gel strength or the bloom strength of gelatin is determined. There are some special types of capsules like enteric capsules which are produced by coating of enteric polymers like phthalate polymers like cellulose acetate phthalate. There might be some sustained release or rectal capsules which are prepared for some special purpose. Then the another important aspect which you should take into consideration is the evaluation of capsules as per the IP and USP standards. The important one is the uniformity of weight. Like if the average weight of capsule content is less than 300 mg, then after performing the test of weight variation or 20 intact capsules, the individual weight should range between 90% to 110% of the average weight. And if 300 mg or more is the average weight of the capsule content, then the percent deviation should be 7.5%. Right? Similarly, the content of the active ingredient in the capsule should also be uniform and within the stated limits of 90 to 110%. Disintegration test is not official for capsules because gelatin shell rapidly dissolves in the stomach. But if we talk about enteric capsules, they should meet the requirement of disintegration of enteric coated tablets as given in the Indian Pharmacopoeia. Similarly, dissolution tests are carried out as in the case of compressed tablet, and the standard apparatus would be employed, right? So the dissolution would be the slowest step or the rate limiting step for the overall absorption determination of the capsules. I hope you like this video. Please click on the like button, do subscribe my channel, and share it on social media. If you want to get the updates about my videos which are uploaded, click on the bell icon so that you can get the notification once I upload a new video. Thank you. Have a nice day.